Hello and welcome to the webinar. Today we'll talk about tRNAs, the rising star of Wobble gene regulators. Among the small RNA classes, such as microRNA and piRNA, tRNAs are somewhat bigger, at about 60 to 95 nucleotides in size. The entire tRNA transcriptome size is about several hundred tRNAs. tRNAs can be processed into new classes of tRNA-derived fragments, or TRF, and tRNA half molecules, or tRNA, with completely different non-translational regulatory functions from parent tRNAs. Mature tRNA sequences is folded into secondary structure resembling the shape of a clover leaf. From the 5' end, it has D loop, anticodon loop, anticodon triplet, T loop, and acceptor arms for amino acid attachment. tRNAs are most heavily modified among any RNAs, with close to 100 chemical modifications. These modifications are an integral component shaping out tRNA structure and functions. tRNAs are tightly compacted into L shape in 3D structure. The canonical functions of tRNA is for protein translation. tRNAs are encoded in nuclear and mitochondrial genomes. tRNA is the most abundant small encoding RNA. About 4 to 10% of the total RNA is tRNA. tRNAs are the fundamental components in protein translation. They are accurately charged with corresponding amino acids by tRNA synthetases. Through anticodons, tRNAs decodes genetic codons to translate mRNA sequence into protein sequence. Among the codons for an amino acid, codon preference and usage reflect protein translational efficiency and accuracy. In the classic textbook understanding, tRNAs are thought of as abundant, ubiquitous, passive mRNA decoders and protein translators. The latest scientific advances have found individual tRNAs are expressed in cells, tissue, temporal, and disease-specific manner. The Wobble effects of tRNA isoacceptors affect translation. Isodecoders can have profound effect on biology and disease. Additionally, tRNAs and tRNA-derived fragments are the most abundant tRNAs in biofluids for biomarker use. For example, the profiles of tRNA isoacceptors and isodecoders repertoire are known to be involved in biological processes such as cell proliferation, differentiation, apoptosis, metabolism, and stress response. tRNA repertoires have dramatic, causal, or associative relations with human diseases such as cancer, diabetes, cardiomyopathy, viral infections, and neurodegenerative diseases. Among the several hundred human mature tRNAs, tRNA carrying the same amino acid but with different anticodons are isoacceptors. For example, alanine has around 50 isoacceptors. tRNAs with the same anticodon but different body sequences are tRNA isodecoders. Here we see each alanine anticodon having several isodecoders. Normally, anticodon and codon decoding is by perfect Watson-Crick base pairing, as shown here, C with G and U with A. tRNAs with this perfect match is called cognate tRNA. However, in ribosome, the third codon base position can have imperfect match with the first anticodon position, such as G with U, U with G, while the first and the second codon base positions are always in perfect match. This Wobble phenomenon is explained by Crick-Wobble hypothesis in 1966 for contorted base pairing or unmodified base by ribosome, and by adenosine to inosine I conversion, not base pairs with U, A, and C. In mitochondria, you can base pair with all 4 bases A, U, C, G, and called super wobble. With the advances of tRNA research and tRNA modifications more known, the modern modified wobble hypothesis incorporates tRNA modifications, particularly at the first position in anticodon or 34th position in the whole RNA, being able to expand or restrict the codon recognition, as shown in the Wobble map in red color. The Wobble effect, or cognate versus near cognate tRNA, is key to the protein translational control. When the supply of cognate tRNA for the codon is greater than the protein translation demand, it is called optimal codon. When the supply of cognate tRNA less than the demand, or codon has only near cognate tRNA, it is a non-optimum codon. tRNA adaptation index, TAI, can calculate the index based on tRNA decoding and tRNA gene copy or tRNA abundance levels. The optimum codon translation speed, efficiency, and decoding accuracy or fidelity are far greater than non-optimum codons. The slow translation, however, helps more accurate in protein folding in the growing peptide chain, and is observed more often in the inter-domain link proteins regions. Not only Wobble and optimal codons affects protein translation, but also affect the mRNA stability. The slower elongation exposes mRNA for degradation, whereas fast translating mRNAs are more stable. Published in Cell, the specific upregulations of tRNA glutamic acid, UUC, and tRNA arginine CCG alone can increase the translation and stability of oncogene AXO-SC2 and GRIP-AP1 mRNAs by their target optimum codons. The increased oncoproteins make the cells highly metastatic and fast-growing in the xenografts model. The tRNA isoacceptor upregulation by either tRNA gene copy number increase or recombinant overexpression can cause tumorigenic phenotype. In neurodegenerative disease, Huntington's disease, huntingtin has a repetitive CAG expansion region, normally encoded for polyglutamine Q. When the charged tRNA glutamine CUG isodecoder level is reduced, such as in Huntington's disease or by artificial locking down, the decoding becomes increasingly prone to -1 friendship to alanine A. The Q versus A ratio in the expansion region determines the huntingtin folding, with more A's causing protein aggregation and dysfunction. This also explains the varying degrees of disease severity. As mentioned earlier, a wobble can happen for unmodified or modified base. In this modified wobble example, MCM5S2 at the tRNA U34 wobble position can preferentially decode DK oncogene mRNA codon usage. DK protein transactivate LEF LIF1 oncoprotein expression, while LIF1 internal ribosome entry sites, IRAS, which leads to breast cancer metastases. Among housekeeping isodecoders for standard protein translation, some isodecoders can perform specialized functions. Published in Science, for example, 1 of 5 tRNA arginine TCT isodecoders is especially expressed in central nervous system. Deficiency in CNS-specific isodecoder causes ribosome stalling on mRNAs in CNS, touches off ATF4-driven stress response, and leads to widespread neurodegeneration if ribosomal rescue factor GTPBP2 is absent. Major human diseases such as metabolic, type 2 diabetes, cancers, neurological diseases, and viral infections by influenza, vaccinia, West Nile, encephalitis, HIVs are correlated with changed tRNAs. At the cellular level, different tRNA repertoire profiles are distinct signatures for differentiating or proliferating cells. The difference in tRNA pools are controlled by tRNA gene expression with the histone marks on the tRNA gene chromatin. As a non-translational molecular function, tRNAs bound to cytochrome C and inhibit apoptosis formation and apoptosis. That is, tRNA is an anti-apoptosis factor. tRNAs are also the first emergency responder to cellular stress, such as heat shock, hypoxic shock, or nutrient deprivation by multiple mechanisms such as A. Reimporting tRNAs from cytoplasmic to the nucleus, causing tRNA depletion in cytosol for protein translation, and B. Deactivation of 3' CCA arm to shut down global translation, and C. Hypoxia-induced tRNA cleavage to form TRF and tRNA fragments, inhibiting protein translation initiation, and D. During nutrient starvation, tRNA modification is reprogrammed to favor translation of protein or enzymes for amino acid biosynthesis. In the past, small RNAs such as microRNAs have been explored as biomarkers. With the advances of tRNA profiling techniques, tRNAs are now emerging as a new class of biomarker. tRNAs have many characteristics that make them a candidate biomarker. They are an abundant small RNA class and particularly abundant in biofluids, even more so than microRNAs such as in saliva, cerebrospinal fluid CSF, amniotic fluids, seminal plasma, urine, and bile. In exosomes, tRNAs are enriched 70 times more than in the cells, way more than microRNAs. The high tRNA abundance means higher biomarker sensitivity. tRNA expression profiles are tied to cell differentiation and cell proliferation. tRNAs display high tissue cell type specificity, spatial and temporal specificity during embryonic development. In human diseases of brain, thymus, blood, spleen, liver, testis or ovary, and breast cancer, tRNAs have shown disease specificity. The sensitivity and specificity contribute to better tRNA biomarker performance. With the exciting new research opportunities of tRNA in biology and disease, Arraystar tRNA qPCR array are designed to profile tRNAs of all anticodons, nucleus, and mitochondria for their isoacceptor families and individual isodecoders. The performance in validated PCR arrays provide rapid, accurate profiling of tRNAs with high analytical sensitivity, excellent even for biofluid samples. In the tRNA qPCR workflow, the tRNAs are first demethylated to remove modifications that inhibit reverse transcription into cDNA. The demethylated tRNAs are copied to cDNA and qPCR quantified using pre-designed primers on the qPCR panel. As tRNAs are heavily modified, the removal of tRNA modification is essential for reverse transcriptase to proceed. Three most important modifications, N1A, N1G, and N3Cs, are removed by ALK-B enzyme. Without such a demethylation pretreatment, tRNA qPCR signals are very poor. With the demethylation pretreatment, the signal strengths are a magnitude higher. In fact, without pretreatment, the cancer and normal samples do not show differential tRNA expression. With the pretreatment, the cancer and normal samples now show very large difference in tRNA expression. The tRNA qPCR panels are compatible with most standard 384 well-played qPCR instruments. Free software tool is downloadable from our website. You simply copy and paste the CD values output from the qPCR instrument to get instant automatic analysis with detailed annotations and useful graphics such as scatter plot, volcano plot, and total differentially expressed tRNAs bar charts. Now we'll talk briefly about tRNA profiling by tRNA sequencing. There are small RNA sequencing kits in the market. However, many of those methods work very poorly for tRNA sequencing due to unique problems of tRNA heavy modifications and tight structure. Arraystar combines the tRNA demethylation pretreatment and also partial controlled hydrolysis to disrupt tRNA structure, also called hydro-tRNA-seq, to obtain much better results. tRNA sequencing covers the whole tRNA sequence regions, including anticodon and body sequences. The tRNA sequencing results are annotated in detail as in the above screenshot. The differential expression is analyzed by Azure using generalized linear models and Bayes statistics. tRNA-seq is the only commercially available tRNA sequencing service in the market. The total RNA amounts required for tRNA sequencing is 5 micrograms. Total RNAs extracted from cells and tissues are okay. tRNAs extracted from serum plasma is not recommended. Indeed, it's better to use tRNA qPCR for low level of the protein, the RNA input. With the tRNA anticodons of interest identified, how do we find the tRNA regulatory target genes? Anticodons of the differentially expressed tRNA isoacceptors can be analyzed by tRNA adaptation index or TAI to compute mRNAs with the most optimum codons, which is based on the tRNA gene copy number, or more accurately, cellular tRNA levels by tRNA expression profiling, by tRNA qPCR or tRNA sequencing, and Wobble pairing efficiencies. Ribosome profiling by RiboSeq is used to know what RNAs are ribosome occupied and actively translated. Proteomic mass spectrometry identifies the differentially expressed proteins. These datasets are intersected to find the tRNA regulatory target mRNAs. In a typical tRNA profiling research roadmap, two groups or conditions such as case versus control, disease versus normal, before and after treatment, with at least 3 biological replicates in each group, are profiled by tRNA qPCR array or tRNA sequencing. The differentially expressed tRNAs are qPCR validated. As a bonus, A tRNA qPCR array is in effect qPCR. A separate qPCR validation may not always be necessary. The differentially expressed tRNAs can be further developed as diagnostics or prognostic biomarkers. tRNAs can be functionally studied by gain-of-function, such as tRNA overexpression or loss of function by lockdown. The mechanism of actions can be studied by finding the target genes by codon usage analysis, ribosome, and proteomics data. Alterations of codon can be used to observe the functional effects and the interactions with ribosomes, translational factors, gene silencing factors AGO2, angiogenin for tRNA fragmentation, white box proteins, or cytochrome C for apoptosis, can provide insight into tRNA molecular, translational, and non-translational functions. Published in Nature Structural and Molecular Biology, here is an actual example of using Arraystar tRNA qPCR array to profile tRNAs to answer an important clinical question: why cancer cells often become resistant to chemotherapy. Many serine and leucine tRNAs were found downregulated during cancer chemotherapy, which is a group of tRNAs that can be cleaved by slough protein 11. Downregulations of these tRNAs leads to reduced translations of serine-threonine kinase ATR and ATM, the central regulators of DNA damage pathway that sensitize the cells to chemotherapy. Slough 11 lockout preserves the tRNAs and upregulates ATR and AMT, rendering cells resistant to chemotherapy and cell growth. So this work has a very significant impact in clinical science. So Arraystar has the tool sets and services for tRNA profiling, either by qPCR or by sequencing. The particularly optimized tRNA cDNA synthesis kit cannot be replaced by other common qPCR reagents. Arraystar tRNA sequencing is currently the only service available that cannot be substituted by regular small RNA sequencing kits. As tRNA modifications are important features of tRNA molecules, with tRNA modification, liquid chromatography LS, LC, and mass spectrometry MS analysis for your tRNA research needs as well. I think I will stop here and will not answer questions as it's pre-recorded. You may now disconnect. Thank you and have a great day.